In this video tutorial, we will be studying about structure of a polynucleotide chain. Now, polynucleotide is actually a polymer of nucleotides. Let's say we have got these individual tiny small individual units. These units are known as nucleotides. Now, if I want to make a long thread or a long chain out of these nucleotides, then that will give rise to a structure like this. This is a polynucleotide. Now, first let us understand the structure of a nucleotide. Where do we get this nucleotide form? Initially, before nucleotide, or nucleotide actually comes from nitrogenous base. They actually give rise to nucleosides, and these nucleosides eventually give rise to nucleotides. It's a two-step process, a long process. From nitrogenous base, something gets added to the nitrogenous base, and there's a nucleoside forms, and then another thing gets added to the nucleoside, which give rise to nucleotides. Now let us see in detail. Namely, there are four types of nucleotides. What are they? They are adenine, guanine, cytosine, thymine, and there's a fifth type also, uracil. This uracil is exclusively present in RNA. Exclusively present in RNA. Now, let us understand how does a nitrogenous base forms a nucleotide. Here there is a nitrogenous base. Let us say adenine is present over here. Adenine actually mixes up with another molecule to give rise to a nucleoside. That another molecule is a sugar. This sugar is a ribose sugar. The sugar has got 5 sides as well as 5 carbons. These are the 5 carbons. Here is carbon, there's the second carbon, there's a third carbon, a fourth carbon. With this first carbon, there is an OH on one side, H on another side. With the second carbon, there is H on both sides. The third carbon has got H on one side, OH on another side. The fourth carbon has got H on one side and CH2OH on the other hand. Now let us count our 5 carbons. This is the first carbon. This is the second carbon. This is the third carbon, the fourth carbon, and this is our fifth carbon. Now adenine, basically what it does, it actually attaches to the first carbon. Adenine attaches to the first carbon like this. It forms a bond with the first carbon. Now, when the adenine forms a bond with the ribose sugar or the C1 or the first carbon of the ribose sugar, this is referred to as a nucleoside. Now, With this nucleoside, another big molecule will come and form a bond. That molecule is a group of 3 phosphates. Phosphate is actually PO4 3 minus. For simplicity, just P has been written over here. So these 3 groups of phosphate, it comes and forms a bond with the 5th carbon over here. The bond looks like this. Now, when these 3 phosphates come and attach with the C5 of ribose sugar, now we have a nucleotide and not a nucleoside. This is known as nucleotide. Now we have got our nucleotide units. Now we have to focus on how these nucleotides are attaching with one another to give rise to a polynucleotide chain. Now, 2 nucleotides, they form a bond with one another and give rise to a polynucleotide chain. Let us say we have adenine nucleotide over here. This is the third carbon. Third carbon has got H and OH on one side. Second carbon. And this is the next nucleotide in line, which has to form a bond with this previous nucleotide. Let's say this is a cytosine nucleotide. This will also have CH2O and 3 phosphates because this is also a nucleotide. This is a cytosine nucleotide. Now, the bond between these 2 nucleotides will form basically over here with O of the previous nucleotide and the first phosphate of the second nucleotide in line. This is how the formation of a bond takes place between 2 successive nucleotides. With this bond formation, when this bond is formed, these 2 phosphates are released. These 2 phosphates are released. Release of 2 phosphates. Now again, a third nucleotide will come. A third nucleotide will come. Let this nucleotide be a guanine nucleotide. This will also have CH2O, phosphate, phosphate, and phosphate. And this will have an OH over here. Again, the bond will form between this O and this phosphate. And these 2 phosphates will be released as PPI. Now, this is a bond. This particular bond, this OPO, OPO, this bond is known as phosphodiester bond. These bonds are known as phosphodiester bonds. So it's the phosphodiester bond which is actually attaching 2 nucleotides to one another. Now, it is said that a polynucleotide chain has a polarity. This polarity is determined by the presence of this phosphates on one end and the presence of hydroxyl on the other end. Let us assume that after this G, another sugar will come another nucleotide is going to come. Let this be another A. CH2O. This is B, and over here another O. This is another phosphodiester bond is formed over here, and that's about it. Let us assume that this is the end. This is the third carbon with its OH and H. This is the second carbon with its H and H. Yeah. Now let us assume that the chain ends over here. This end over here will be referred to as 3' hydroxyl end. Why? Because this is the third carbon, and third carbon has an hydroxyl attached to it. That's why this is referred to as the 3' hydroxyl end. Now, the starting end is known as 5' phosphate end. Quite obviously, because this is the fifth carbon and there is a phosphate group attached to it. That's why this is referred to as a 5' phosphate end. Now, this is a polynucleotide chain. That's how the structure of a polynucleotide chain looks like. It starts from over here with 5' phosphate end, because before this nucleotide there is no other nucleotide. Had there been a nucleotide, then that would have been the starting point of a polynucleotide chain. Now the starting point of a polynucleotide chain has a 5' phosphate end. After this particular nucleotide, the second nucleotide is joined with the previous one with a phosphodiester bond, with the fifth carbon's hydroxyl getting attached with the phosphate of the second nucleotide, and every time the same thing continues, and these bonds are known as phosphodiester bonds. Now, the terminating region where the chain would end, no further successive nucleotide is there to form a phosphodiester bond. That's why this hydroxyl will remain as it is. This OH will remain as it is. This end is referred to as a 3' hydroxyl end. That's how the structure of a polynucleotide looks like.